Hi everyone, welcome to another session of the AFMS seminar series. Today, actually, I'm hosting the Professor Nam-Trung Nguyen from the Griffith University, and he's gonna just present macro elastofluidics, elasticity, and the flexibility for efficient microscale liquid handling. Thank you so much for accepting our invitation, and we are all ears to listen to your presentation.
All right, thank you for the introduction and thank you for inviting me for this seminar. Um, as you see, I'm from Griffith University. Previously, I was the director of Queensland Micro and Nanotechnology Center. And since last year, the center merged with the Center for Quantum Dynamics, and now we are called Queensland Quantum and Advanced Technologies Research Institute, Quad3. And I'm now in my second year of my laureate fellowships. So broadly working on the topics I'm talking about today. Now, let's go to the motivations of what microelastofluidics is. There are few motivations. I have three motivations for research in this area. The first one is now very fundamental, looking at fluid structure interactions in the time and space map. So you know that thing happen in physics. Our current physics happens in space and time, and then in space we have the length scale, and in time we have the time scale. Conventional fluid structure interaction in what you are maybe familiar with in turbulence flow has time constraint and also side constraint due to the eddy size, for example. Now we try to look at this from a very broad perspective and see in this space of length scale versus time, where are the interaction? What have we, say, neglected so far? And what can be done in these empty spaces where there's no activities? Right, so you look at this, in this space, we have all the line which are length scale divided by time scale, which is speed of things, right? So we have basic speed of things such as speed of Brownian motion, speed of sound in air. So if for people working on turbulence, now speed of sound in air is, now this is where the Mach number is calculated, is where you also determine the physics of the interaction. Then you have speed of sound in solid, and the ultimate speed, what we know in our classical physics, is the speed of light. That means beyond of that, there's no physics, no interaction we can obtain, according to classical physics. Now for what I'm doing, I have been working on microfluidics. That means working on handling liquid and looking at physics of liquid flow or fluid flow in the microscale. So that's why I listed up the length scale, say below 1 meters, but typically microfluidics or microelastofluidics devices or phenomena happens in the scale of 1 millimeters or below, right? So 10⁻³ of meters. So there's the blue area. You can see the broad blue area. And in conventional microfluidics, time scale can go all the way down to 100 microsecond. That's the fastest you could do. And what we did is the physics in this area, in these blue areas, actually is the physics we are currently working on. So think we can manage with our micro technology, making devices down to micrometers or smaller than micrometer scale, and working with interaction that happens now faster than 1 millisecond or 1 microsecond. And you have all these phenomena like viscous capillary resonance, capillary inertial resonance. We also have mechanobiology thing, interaction with cells, which happen in the same small scale but much slower. And if we go further down with the time scale, the speed and the time scale are the speed are so fast and the time scale so small that the kinetic energy of things are large enough that we can even look at molecular interaction. So in this case, the energy induced by fluid structure interaction can help chemical reaction to overcome the energy barrier, and then we come into an area where fluid structure interaction can enhance or enables chemical reaction. So we call it sonocatalysis. Is the area we can also touch on with the small scale of devices we have and with the fast scale of the interaction. And for all this area, you can see the blue, red, and yellow areas with acoustic wave in solid, for example. We all can say technically touch them using state-of-the-art technologies. Right, so that's the motivation of why we are looking at from the big picture what could be done with microelastofluidics in terms of fluid and structure interaction. Now, the second motivation is the practical one. So we know that there is an emerging area of wearable devices where flexible devices are made for wearable on skin or implantable in the body. So we have a range of sensing devices, drug delivery devices, which are flexible. Now, in all these devices, there is a missing mixing functionalities that is the fluid handling part. So most devices are made of electrodes. You can collect signals, you can send in electrical signals to nerves, but the development of fluid handling capabilities are still limited. So we still need to have a lot of things like mixing, filtration, separation dosing, storage, and actuation with conformal attachment because biological systems are soft and have different shapes than solid system on the bench. So to deliver or to provide this functionality, we need to have another approach in terms of developing microfluidic devices, right? So we need to revisit the entire range of devices from now liquid deliveries, mixing, storage, actuation, and so on. So that is very practical motivation. And the third motivation, the actually more systematic one, which provide a more smooth transition from conventional microfluidics into the field of microelastofluidics. That now allows you to get straight into the field now without much investment of how to get a small space, how to get a fast interaction. And this schematic actually is based on conventional microfluidics. That means microelastofluidics can have two areas. One is a digital microelastofluidics, and the other is continuous flow microelastofluidics. Now, in digital microelastofluidics, is similar to conventional microfluidics, where you deal with droplets as a digital discrete entity for handling liquid. But instead of handling liquid itself as a droplet, now we make liquid becomes now solid-like. So we make it flexible, solid-like, so that we can handle them like handling particles. Right, so if you have existing technologies for handling bits, particles, now we can apply it to liquid as well. If you make a liquid, droplets become solid. So we can do that by making liquid marbles coated with small particles. So making the liquid droplet not wetting, or coat the liquid droplet with a solid polymer capsule. So it's like a liquid bits, or you store the liquid in a hydrogel matrix. So you make it hydrogel bits and handling all this platform as you are handling solid bits, right? Now, on the side of continuous flow microelastofluidics, you can also utilize the fluid structure interaction, the flexibilities of things at different scale and utilize them for microfluidic functions, right? So at the molecular scale, simplest thing is looking at flow with long chain polymer mixed into it. So the liquid becomes viscoelastic. And at the low Reynolds number, normally you won't expect to have turbulence flow because friction force is much higher than inertial force. So we forget about inertial forces, but with the coiling and recoilings of small molecules, you can have a so-called elastic turbulence, which again can have a local turbulence effects, and that lead to a number of applications as well. You can use it for separation, mixing, and so on. So this is a molecular scale. Now for the device scale, fluid structure interactions, deformations also can give you direct tunabilities of functional devices. That means previously rigid devices need to need to go through design iterations to get the right parameters. Now giving the devices the flexibilities or the tunabilities of dimensions or other functionalities, you just need to make one design and tune it with stretching or compressing, for example. So I will give some of the examples here from our recent works to show you how it works. All right, so just go through all this platform and their application. The first one is liquid marbles. As I mentioned, to handle liquid as a solid, we just make them solid. Now the simplest way to do it is coat the liquid with hydrophobic powder, and the hydrophobic powder actually isolate the liquid to the surrounding, make it non-wetting. So you have these so-called liquid marbles. Now the advantage of the liquid marbles is behave like an elastic balloon. So you can squeeze it, and then when you release the stress, liquid marble come back to the original form. It's like water balloon, kids playing with, and that allows us actually that allows this platform to be very robust in handling. Right, it's not like a liquid droplet when you put it on the surface, either it's roll away if the surface is super hydrophilic, or it's just wet and go somewhere. Liquid marble will stay in its form and the content is protected. And we have characterized it, and now it has very interesting elastic properties. So you can squeeze it and release the stress. The liquid marbles come back to the original form. And this stability allows us to handle complex things such as 3D cell culture. So in this case, the non-wetting behavior of the liquid marbles also allow cell not to attach to the wall of this droplet and force them to form so-called spheroids. Right, spheroids are cell group, or sometimes we call it organoids. If you co-culture many cell type together, and we use it as a 3D cell culture platform to form spheroid of olfactory and shedding cells that are cells from the base of the nose. And this cell has the properties of helping nerve cell to grow back together. And this potentially will be a way to treat spinal cord injuries. So you get nerve cell to grow back together, and to have this therapy done effectively, we need to have healthy spheroid of these olfactory and shedding cell to implant back into the body. And the liquid marbles or floating liquid marbles platform allows us to grow this healthy spheroid, which is now healthy from the core to the outside. And because cells come together naturally, the density of the spheroid adjusts by itself so that the core is not dead, so that the whole spheroid is healthy when you implant it in the body. There's one application. Now the other approach of making a liquid droplet solid or particles is encapsulating liquid in a solid shell. And this method actually used the traditional microfluidic, droplet-based microfluidics techniques. So what we do is we form doped emulsions. So a droplet, two-layer droplets, where the outer layer is UV light curable polymer, or the outer liquid can be alginate gels, where you can solidify the alginate using calcium ion or other metal ion as well. So this approach allows us to encapsulate liquid inside a hard shell, and then the subsequent processes are dealing with the liquid as dealing with the bits, or solid bits. And there are a few applications for that. So you can see here, we can also make a liquid bit with an aqueous core. That means water-based core. That means all chemical reaction or all analysis we have done so far in a tubes can be packaged and done inside a single liquid bit, as you can see here. And this platform, again, if you make the encapsulating polymers porous, like using an alginate hydrogel, this can serve as a platform for slow release of drug as well. So encapsulate drugs inside polymer shells, and these bits now size is about 50 to 100 micrometers, and you can use it as a slow releasing platform for drugs. Same thing for environmental applications. So we use the same concept, but now we don't encapsulate liquid, we encapsulate cells entirely in the gel matrix, in the hydrogels. And in this case, we encapsulate microalgae. So the interesting thing is, once encapsulated, microalgae continues to grow inside this hydrogel matrix. And the application for that would be environmental repair. That means if you have microalgae in these bits, you put it in water which are contaminated with nutrients like nitrogens or phosphorus. The algae will grow, consuming all these contaminants. And once the algae fully grow, you can take them out, right, and this is discarded. Another application is growing algae. So say the edible algae in this platform. So you can grow it into a full size or package in hydrogels, and you can use it as a food stock, either feeding animals or for consumption by humans. You know that microalgae is a superfood, very healthy. So this is another application of making liquid solid. Now, another more practical or interesting application of this platform for analytical chemistry is digital polymerase chain reactions, or digital loop mediated isothermal amplification. So because DPCR or DLAM. So the concept is to encapsulate a single DNA samples inside this bit and run the amplification. And because of the small size of the bits, some particles or some bit will contain the DNA, and some may not have the DNA, right? So if a bit contains DNA, you run it through the amplification, you will have the bit light up with all the DNA amplified. Those have no DNA will stay and remain dark. And instead of measuring the intensity of the fluorescent signal, now we just need to count the number of bits that light up. Right, I can see in this this graph for PCR for PCR and LAM amplification, that the number of bits which are light up are the bits which contain DNA initially, one single molecule of DNA, and is correlate very well with the actual concentration we know. So the test showed there's a linear relationship for both PCR and LAMS. Now, conventional digital PCR are done with droplets, so you need to form a very stable emulsions to do the same thing. But now, because we encapsulate the liquid, encapsulate the samples inside a hard shell, so it's now it's absolutely stable. That means you can take it out, shake it, and the bits will not merge together because now the solid shell protect the content. It's a very reliable method for digital LAMS and digital PCR. All right, now we move further to explore possible application with these liquid bits. One way is we have to coat the bits with something. Either you make it fluorescent, right, to make it look different to detect and maybe sort later on, or you can make it magnetic. So now using magnetic force to sort and separate bits. So in this case, we just mix the shell polymer with magnetic particles. So that means we make a magnetic liquid bits, which is responsive to magnetic fields. And we demonstrated with separation experiments. We mix up bits, one with normal shells and the other with magnetic shell, and you put a magnet next to it, and those with magnetic shell will be diverted to the lower branch where the magnetic force pull them to the side. So there's a demonstration on how to labels or how to address this liquid bit platform for further processing. So magnetic force is an example. Now another approach is using dielectrophoretic force, which is much easier than magnetic force, because all the bits or all the particles have the dielectrophoretic footprints or characteristics just based on their geometry. Because the core and the shell have different permittibilities, dielectrophoretic properties. Now, if you have bits with different core and shell geometry, they respond to a electric field differently. And using this difference, we can also sort out bits with various geometry. So for example, a solid bit with no liquid inside, a liquid core solid shell bits, or liquid core solid shell bit with different core sizes, all respond to electric field differently. And with that, we can also sort them in a microfluidic system. Right, so that's all possibilities we could deal with this platform. Now, just a brief touch on fluid structure interaction in the molecular scale. I mentioned before there are now micro viscoelastic phenomena we could deal, but we also looking in the area where I just mentioned that the fluid structure interaction in such a short time and short length scale allows the kinetic energy to be on the order of magnitude of the energy barrier for reaction. So all we need to have is to create small air bubbles and induce cavitation in these air bubbles. So excite them with high frequency, and bubble collapse will create high pressure and high temperature that allows chemical reaction to happen. And that's called sonocatalysis. Is an emerging area. And we did it either by using microporous materials where the pore has air pocket, and this air pocket are excited with an ultrasonic field to induce cavitation. Or we produce, as the setup down here, we produce air bubbles in a microfluidic device and excite the whole system with high frequency. So the air bubble oscillates and induce gravitation inside them, and that also produce reaction that normally not happening. So that's an emerging area. There's still a lot of things to explore, and probably this is a new direction for chemical reaction of, say, green sustainable chemical reactions, because the energy you put in is not a chemical energy, is a mechanical energy, right? Just ultrasound excite thing and then come over the energy barrier of reaction. Now the next example is the device scale interaction. That means we want to look at the formations of flexible system and see how can it control flow, or the other way, how flow can deform thing and then result in functionalities that we cannot do not otherwise. So that is an example of a passive flow controller. So in a way, this is also called a fluidic transistor or a self-tunable micro valve. So in the left corner, you see the concept, how the device works. So we have a flow channels, and then you have a membrane, which usually works as a valve. But the membrane deform if you have a high pressure, inlet pressure, right? So normally, the higher the pressure going in, the higher is the flow rate. So you cannot cannot restrict the flow rate. But with this structure, the membrane deform and constrict the channels and limit the flow rate. That means after initial pressure range, flow rate will remain constant. And that's called passive flow controller. And that, again, is a very typical examples of fluid structure interactions application in microfluidics. And we can model the whole things in COMSOL using structural deformation and fluid flow simulation. And here, for example, it just shows the simulation results versus the experimental data. So that means you can increase the pressure to a certain threshold, and from then on, the flow rate is kept constant. Okay, so for many, for many applications, there's a simple solution for flow control without external actuators and flow sensor, right? So you just design the device for a fixed flow rate, and doesn't matter what kind of pressure you apply on, you always get a constant flow rate out of the device. Now another application of this fluid structure interaction is now in a smaller scale, is surface microfluidics, where the surface properties or the microscopic surface properties can be tuned by stretching. And that has applications in cell culture or in mechanobiology. That means we can induce stress on cells and allow them to differentiate according to the mechanical cues. So a very simple example here is we make an array of pillars on a flexible substrate, PDMS, and then stretch them. All right, stretching actually changed the topology of the gap between the pillars. And of course, as expected, the change in morphology lead to change in surface energy or contact angles. I mean, if you put a droplet on top of this array, stretch them, the contact angle of the droplet will change as well. So this stretching of flexible substrate provide another means of controlling surface energy. And that has application, for example, in controlling the fate of stem cells if you grow cell on it. And now stretching the substrate to change the surface energy of the hydrophilicity of the surface and make cells respond or react accordingly. Right, so another applications of stretching is tunable microfluidic devices. So for many microfluidic applications, in this case is initial microfluidic separation. To get the device change the characteristic, we need to change the entire design. And by stretching a flexible device, now we can tune the geometry down to micrometers scale, because when you stretch, the strain is constant. So say 4% of stretching of the overall device means you stretch by a few millimeters, but in the microscopic range of the features inside the device, you can tune it down to 1 or less micrometers. And that give microfluidic devices now an opportunity to fine tune their properties. So in this case, for example, initial separation, stretching change the channel length. And with the channel length, you can change the focusing position of particles or cells. So you can see in this slides that larger particles or larger cells is focused in the middle, smaller one go to the side. And by stretching the whole device, you can tune it to the center of the device and separate the larger particles now through the middles of the device. So that here is in video. Maybe it's not clear enough, but here we use it for separation of cancer cell from whole blood cells, just by tuning the length of the device. Well, based on the same idea, so stretching, macroscopic stretching allows you to control a very small, minute length scale in the actual microfluidic device. And here you have a horseshoe structure which trap particles. And the video show you when the device is stretched laterally, and accordingly the cap between the horseshoe structure just open up a little bit one by one. And with that, you can release particles which were trapped there before based on their size. That means now the cap is open step by step to release particles now of the different sizes. So we use it to trap and release particles according to their sizes in this kind of array. The interesting good thing is now the stretching in macro scale could be controllable in a millimeter scale, right? So you can stretch few millimeter with a precision positioning system, but the accuracy you can get for controlling the cap here can go down to a few micrometers or even smaller than micrometers. So very precise tunabilities. Right, so next one, applying the same concept to conventional droplet-based microfluidics. Now, instead of, you know, that for droplet formations, we need to adjust flow rate, we need to adjust channel geometry to get the right size of the droplets and also the frequency, how the droplets are formed. And instead of changing all the geometry, now we just need to stretch it, to change the geometry by stretching. And the trick of controlling the stretching is we make a flexible device, and in places where we don't want to have change during stretch, we make it extremely thick, right? We have all this PDMS block placed on top of channel structure, which we don't want to deform. Only areas which we want to deform and to control will be a thin layer, right? And with that, we can selectively control the geometry of T-junction or co-flow junction where droplets are formed, while we're keeping the channel geometry elsewhere constant, right? So that's just a basic idea, but then again, this can apply for droplet formation, or it can also apply for the formation of dopal emulsions. You see the application of these chips for making liquid bits and all the application before. And for dopal emulsion, we have various junction which control the size of the core, the thickness of the outer layer, or thickness of the bit, and also a junction controlling the distance between the bits, or distance or the frequency of the bits. So we have here, for example, 3 junction, which we want to control separately. So all we need to do is, if we want to control junction 1, we make it flexible and make junction 2 and 3 rigid. And the same thing, can do it to junction 2 or junction 3. And with that, instead of making a complete new range of devices, we just need to have one device and tune the results, tune the now the shape of the dopal emulsions and the size of the dopal emulsions just by stretching, by tuning it. Now, applying the same concept again, other droplet manipulation tasks can also be done with this stretching mechanism. For example, when droplets are formed in a microfluidic system, you can split them into 2 daughter droplets. And normally, the droplet will be symmetric if you split them at a T junction and both arms of the junction are the same, or say have the same hydraulic resistance. Now, stretching can change the fluidic resistance or hydraulic resistance of these branches, which in turn change the size of the daughter droplets. That means now we have a tunable device which can decide how a mother droplet split into 2 different sizes. And that also, now this is another, this can be achieved on a single device, on a single design. You can see on the left corner, bottom corner, how strain or stretch can vary the volume ratio of the 2 daughter droplet. So previously we have to use either heat or magnetic force or electrostatic force to control this process. Now we just need to simply stretch it, with a certain strain. Now another application of this stretching method is tuning the merging and mixing process of 2 droplets. So that is also a very basic process of droplet-based microfluidics, where we need to trap 2 different droplets, A and B, merge them, mix them, and then let chemical reaction happens in the merged droplets. And as the stretching concept again allows us to control this process. That means we can slow down droplets, make one droplet, wait for the next one. You can see in the middle, the blue droplet has to be trapped, wait for the yellow droplet to arrive, and then merge them together and move them on. And all this can be tuned just by stretching the merging chambers. So that's latest work we have done in this series of droplet-based microfluidics, tunable by stretching. All right, so another and maybe last application of all this concept is inducing stress and strain on cells, right? And then observe the expressions of a certain biomarkers. An application for that is early cancer diagnosis, right? So the basic concept is we take a samples, tissue samples, which normally we cannot discover whether the tissue is cancerous or not, because at the early stage, the cells do not express the biomarkers typical for cancers. Now, what we do now is we take this sample, put it on a stretching device, make the cells exercise. Now we stretch them periodically, and after a while, cancer cells typically respond more strongly on all these mechanical stimuli and express the markers strongly. So, and we test it with clinical samples, I mean, the real cancer cells of different stages. And clearly, by stretching them in this stretchable microfluidic platform, we can see clearly the expression of biomarkers which tell that the cells is cancerous. So that's now is an option for early detection, that we don't need to wait for the cells to become really cancerous to know that there is a potential of cancer from these samples. All right, so that's a few of the examples. You see the big pictures of what we meant by microelastofluidics, what can be done if we look at fluid structure interactions in microscale and in a small time scale. There are lots of things still to be done there. And very simple application, as I demonstrated here, is just to look at the deformation, flexibilities, interaction back with the fluid flow in the microscale already result into many interesting applications. One thing I haven't presented here is using all this concept for wearable devices. That means for flexible devices that you stick on skin or implant in the body, where you still have sensing functionalities, fluid sampling functionalities, mixing, and yeah, so there's our ongoing works, and I hope to present in a later time. But what you have seen now are actually now a lot of application we can now we can discover with this basic concept. And I hope that this talk will give you some inspiration on what we could deal with with this type of interaction for your own research. Thank you.
Thank you so much, actually, to give this great and informative actually presentation. It looks very informative to me. And you did try to introduce different examples and application of the microelastofluidics to the audience. So if we have any question from the audience, please speak up or unmute your mic, or even you can just type your question in the chat box and I will ask the question. Any question?
Hello, can I ask a question?
Yeah.
All right, great. Thank you very much, Professor Nam-Trung, for the very inspiring presentations. I know that the video presentation will be online, but I can't help screenshotting many of the slides. I think one question is related on the controlled particle release depending on the size. On slide 25, where you stretch the microfluidics and then the particle of different size are released. I'm just wondering how those small particles are initially deposited on those cavities, which is not shown in the video.
Yeah, you flush the samples through the chambers, and those trapped into the horseshoe structure will trap there, and those not will be washed away. So that's where we start with. So
Okay, I see.
We start with the structure which already have particle trap on it.
Okay, I see. Got it. And then basically what we are seeing is that in all those cavities, the particle size distribution will be similar, but the subsequent flow injection, and then depending on how much you stretch the microfluidics, can kind of selectively release the particles, right?
Yeah, so when you stretch, you practically adjust the cap of the horseshoe structure.
Yeah.
If the cap is big enough for certain particles, they will pass through. Right,
Okay.
and the chairs will latch that. They will flush the small particle out first.
Yeah, got it. Thank you.
Any other question from the audience?
Can I ask a question?
Yeah.
Thank you, Professor Nam-Trung, for your excellent talk. I've learned a lot. I'm curious about for using viscoelastic fluids for mixing what polymer concentration and molecular weight you are looking into. So are you using the channel structure for mixing, or you're curious about using the second normal stress or other properties for mixing?
I don't have it in my mind, but this work was done probably 20 years ago. So we use this, you can see on this slide in the middle, there is a sudden expansion structure.
Yeah, yeah.
Where you induce less share at the junction.
Yeah.
And for my understanding, the concentration was dilute because you cannot afford high concentration because viscosity is very high. You need to have a high pressure to push it through. Yeah, I have to check in the paper. You can also check. We published it long times ago.
Yeah, I was wondering like if a higher concentration and molecular weight would have a more enhanced turbulence.
It will have more enhanced turbulence, but you'll have big challenge in making the device. So PDMS device will not work in this case because the pressure is too high. It will
Yeah. Right.
Yeah. So we need silicon and glass. So you need to make the device in silicon and glass, and you can have highly viscous liquid passing through. Yeah.
Okay, I see. Thank you.
Any other question?
But then no, we haven't explored. You can use diluted polymer solution and combine it with the stretching approach as well. So we tried with stretching before, just using Dean vortices, right? Because when you stretch, you can tune the Dean vortices as well.
Yeah.
But diluted polymer solution, you have combined with Dean vortices by advanced mixing as well.
Yeah, that's a good point.
Yeah. So don't need to have highly concentrated solution.
Thank you so much. While actually you don't feel very well, Professor, you deliver the excellent presentation. Thank you so much. And once again, thank you so much for accepting our invitation.
Welcome.
Appreciate it.
Right. Thank you everybody for joining. I need some rest. I have terrible flu. I don't know whether it's COVID or not, but I'm at home.
Yeah, hope you feel actually very well soon. Okay, thank you. See you everyone.
Thank you.
Thank you. Bye.
Need to see how to get out here.